Good evening, viewers. Now is the second session of our biological series. Today's topic is on immunohistochemistry. What's in the picture? The temptation of image manipulation in research by our speaker, Dr. Avijit Banik. Dr. Avijit Banik is a cognitive neuroscientist with specialization in the field of Alzheimer's disease. He obtained his PhD from PGIMER Chandigarh in 2014. He joined his first postdoc training in National University of Singapore. Currently, he is working at Emory University, Atlanta. He is well versed with molecular genetics, cellular biochemistry, pharmacological assay associated with drug discovery program, and is an expert in behavioral neuroscience. He is a dedicated scientist and has always been an inspirational trainer for his fellow colleagues and junior students in the lab. Recently, his department at Emory School of Medicine nominated him as a COVID-19 lab hero for sustaining essential work during pandemic. He has more than 20 peer-reviewed publications. He is actively involved in current Indian science technology innovation policy formulation, being an active committee member of an US-based non-profit organization, Science and Research Opportunities in India. I'm pleased to invite Dr. Avijit for his presentation. Over to you, sir.
Thank you, Parul. You guys can hear me?
Yes, sir.
Okay, great. Thanks, Parul, for the kind introduction. And like being the alumni of NRL, it is always pleasure to come back and, give this kind of, pep talks on different research methodologies. What I used to do when I was a senior demonstrator in NRL under the leadership of Dr. Akshay Anand. So I love teaching. I love, training students. I have been doing so here as well in Emory University, training a couple of undergrad students, and it's wonderful. So I was told by Dr. Akshay to choose something which probably not too much into the depth of the research what I am doing, because I am a neuroscientist and I am working right now on Alzheimer's disease drug discovery program here at Emory University Pharmacology Department. But considering the viewership of this particular series for biology, I thought to choose a kind of a generalized topic, and that is probably most important and significant, and it's very critical also in day-to-day research life. So basically today's talk is how you should, resist from the temptation of manipulating images in the research labs, and also a little bit of part of the research, image quantification using images software that will be coming into my later half of the presentation. So without delaying much, so if you just look into that title image, it's that itself says what exactly is the image manipulation. That from outside what you see and from inside what you see, probably just juxtapose what is there from another image, and eventually you get what you desire for. But yeah, eventually you end up doing the research misconduct. So you have to refrain from such. And okay, yeah. So what is the definition of research misconduct? As per US federal policy, research misconduct is defined as fabrication, falsification, or plagiarism in proposing, performing, reviewing, or in reporting research results. So now the image which are also coming under the purview of the research data, even the image manipulation also comes under research misconduct. So now there are certain limitations, but certain approval also as far as the research, the image manipulations are concerned, but you need to know what exactly you can do and how far you can go. So it is now very simple, and there are so many sophisticated softwares and freely available softwares that they are like Photoshop, Image, and that's how you easily get tempted to beautify your images from your research outcomes. And that's how you try to manipulate the constitute or inappropriate changes into your original data, and that can be classified as well as scientific misconduct. But you have to be cautious that there are skilled editorial staffs which are available in different editorial houses. Whenever you are submitting those images for the publication, they can easily identify those manipulations and raise the alarm bell for you. So you need to know what exactly is uh, uh, approved and what you cannot do. So as per the journal guidelines, in 2004, the first time in Journal of Cellular Biology, the uh, the editor came up with a kind of a scientific image manipulation related guideline. And since then, there are several other uh, uh, other journal houses. They have also taken this into consideration. But so far, not all of them have made it mandatory. And unfortunately, in all of these journal houses, the guidelines are not widely comprehensive. And you can see there are several I have listed down here, like Journal of Cell Biology, Nature, PNS, Science, JNS, and the Journal of Cell Science, Molecular Cell Biology. They do have certain guidelines in place if you just go through the guidelines for the authors. Now, why it is wrong to touch up the images? You certainly don't want to misrepresent your data, isn't it? So you are deceiving actually your colleagues who expect and assume the basic scientific honesty that whatever you are publishing, it's on the true nature, and they don't expect that you have hidden some facts from your image data. Now, manipulating images to make figures more convincing may also deprive others of seeing critical information that is often hidden in a picture or other primary data. And that's how it is very difficult to reproduce the data coming from one lab and reproducing it from another labs, because we always try to tend to beautify our images, and by doing so we eliminate critical information from the images, which probably could be very, very useful for other research lab to follow. Now, being accused of misconduct initiates a painful process that can disrupt one's research and career. As you understand, there is a threat involved, and you certainly don't want to be on the wrong page. It is also important to understand that when ethical lines are drawn between acceptable and unacceptable images, that you should be always at the acceptable side, but not on the unacceptable side. There is rampant rise in research misconduct in the recent years while falsifying the images, and there is a research integrity body in US who every year take up 20 different cases related to research misconduct. And during 2007 and 8, they found 68% of those cases were related to falsifying images, and that has increased from 40% to 68% from previous 2 years. Whereas in 1989 and 90, this percentage was very much lower, but probably that time they were not thoroughly checked. And now as the year progresses, the number of publications are also rising, different journal bodies are there, and so one has to be extremely careful and critical that what you are trying to publish. And you already know that there were certain allegations last year in June from CSIR IITR that almost 150 different publications were flagged off because of that image manipulation, where the images were duplicated or altered or otherwise manipulated to distort the results, and most of them were coming from Western blots and microscopic images. Now, recently in January 2020, the Director General of CSIR, Professor Shekhar Mande, he has investigated thoroughly into those cases and submitted the report to respective labs. And after that, I really don't know what actions were taken. But yeah, those are the serious misconducts if proven and found guilty. Now, in next 3 or 4 slides, I would like to show you that what exactly the image manipulation looks like. And mostly if you see that in the blots where like you run a protein, like in the Western blot in the jail, and then you image them into into an imager, and then you want to publish that, and once again you want to beautify that in a way like you want to delete certain band. And if uh, you see the in the original image in the red circle where that band was present, whereas in the manipulated image the author have removed that. And it is very easy to do that, but it is also easy to identify also by the publishing house. Now in the lower panel you see they have added a band to a blot, where in the existing original one there was no presence of a blot here, sorry, presence of a band here, whereas they have added uh, superficially on top of that. So these are all termed as serious research offense, research misconducts. Now in the blot manipulation, uh, authors also try to tend with brightness and contrast adjustment. So in the first panel, you see they have adjusted the intensity of the single band to show, I mean, to disappear, to basically to eliminate that band from that gel, as well as they have also adjusted the contrast in image panel B, so much so that they have actually eliminated all the backgrounds, and in panel 4 basically it's like totally ended up with a single very beautiful band of a particular desired protein, let's say. So in a way you are actually falsifying the data, that although in your original blot there are several other protein molecules or peptides are present, whereas in the manipulated image you have tactfully eliminated them. So those you have to be very careful to refrain from doing so. Now coming to the microscopic images, many a time you see that the microscopic images from the immunohistochemical analysis, where the tissue sections are taken and then finally the images are captured, and then those images are further modified for your requirement. Now in this particular image you see the intensity of the gold particles around the cells are manually filled by black color. So in the original one you see they are little faded, but in a manipulated one they were very dull. Now is that wrong? I would say yes, because you have specially introduce the intensity on a certain part of your image, which is not acceptable. If you want to change the intensity of an image, it has to be in a whole way, holistic way, but not specifically to those certain dots. Now there is a way you can also color those particular dots, or maybe you can arrow them to direct that into that. But here author have specifically uplifted the intensity of those dots, which is not at all acceptable. Now misrepresentation of the microscopic field, what did they do is here, like in the manipulated image you see so beautifully cultured cells. I don't know which cell type they are, but they looks really nice, like with all the cytoplasm and the nuclear body intact. But when that manipulated image were checked for contrast adjustment by the scientific body, it was seen that these particular three which are coming with a black background, they actually juxtaposed into this particular image. So basically this view is not coming from a single microscopic view, but couple or two or three more images were combined together to give you a kind of a feeling that okay, there are so many cells in that microscopic view. This is absolutely no, you cannot do that. Now coming to the intensity, you can certainly play around with the intensity as long as you don't eliminate the true sense of your finding from the images. Now let's say in this one, the left one is the unprocessed image, and the middle one is the appropriately processed image, and the right one is like too much over processed image with the intensity. Now what happened, you see uh, on the uh, you know uh, on the histogram, you see from this image to this image, although this is modified, I would say it is manipulated, but it is acceptable because you see the histogram graph is not changed from this image to this image. But whereas in this image, because it's too much intensified, this histogram pattern is completely changed. So which is basically changing the original information from the image. Now when you are trying to focus into a certain region, you see in the small boxes here, now in between unprocessed versus overprocessed, you look at the look at the histogram again to show that how they are changing in between unprocessed versus the processed image. So now once you try to represent this particular figure instead of this or this, you are basically manipulating the values from those, and which cannot be accepted. To learn a little bit more about image resolution, that what is the resolution in an image is basically the dot per inches, which we call in short DPI. And most of the journals, they require at least 300 pixels per inch, which is called 300 DPI, for these digital images to publish. Now, you have to understand that when you are generating these images, let's say from the microscopic view, they will have certain pixel sizes in them. So now if you look into this particular image, like let's say this is the original image of a small fish, and now once you enlarge it, you see those small boxes which are nothing but the pixels. Now 300 pixels per inch means for an image in a width and breadth, you will have 300 pixels per inch. So nothing but in a square inch area, you will have 9,000 90,000 pixels bands in that. So you have to understand that when originally you are capturing the images, if it is not coming by 300 by 300, then manipulating that is also sometimes changes the overall composition of the image. Now it is also always referred that you should record your images on TIFF format, because in TIFF the file compression goes lossless. So lossless means like there is no loss of any pixels. Now in JPEG or other formats, when you try to compress that image, you lose that pixels, and eventually it alters the spatial resolution of the image. So that's why almost certainly all the journal houses, they tell you to submit your images in TIFF format, but not any other formats. So now this is a kind of an ethical guidelines published by the Science and Engineering Ethics journal. And whatever I have discussed so far, they are basically pointed out here. Like the scientific digital images are the data that can be compromised by inappropriate manipulation. Manipulation of the digital images should only be performed on copy of unprocessed image data that you have the original file, you have done a couple of times processing, and then further you want to represent that. No, it cannot be. All the processing should be done on the raw images. Obviously you save the raw images for your further or future purpose, but eventually you have to use those. Now simple adjustment to entire images are generally acceptable, like you can crop it, you can resize it, but just make sure that while resizing you are or like when you are magnifying the image, make sure that the resolutions are in the acceptable ranges. Be careful when changing the size, because in that doing so you are also changing the pixels. So and most of the times you need at least 300 DPI, 300 pixels in that range in order to submit those images for the publications. Now, digital images that will be compared to one another should be acquired under identical conditions. Now, let's say I want to do certain intensity changes. I certainly don't want to do only to my test samples, but not to my control samples. So in a way, you are skewing your test samples, but in the control samples, you are not doing any changes. That is not acceptable. Now, secondly, you are not allowed to do also part alteration in your image. Like let's say in a certain region of your image, you want to manipulate that intensity. No, you cannot do. Overall, you have to do that for that entire image. So these are all basically whatever I have just discussed so far. They are basically, put down together as a kind of a guideline. And uh, this is how you should practice. So you should know and understand that to what extent you have the limit to beautify or modify your images, and while doing so, make sure you are doing it thoroughly overall for all the images together. Now in the second part, I would briefly explain about like how to quantify the histochemical images using ImageJ. So here in Emory, me along with a couple of undergrad students, we have come up with a automated process of image quantification using ImageJ. Many of you might have known, or already might have used ImageJ. It's a wonderful software. You can do lots of analysis there, quantification and counting the numbers. So in particular, this slide, you see this is a kind of a primary microglia cell, the BB2 cells, and sorry, yeah, and those orange dots inside are nothing but the latex beads. So this is a kind of an image to show how cells phagocytose. So you put those beads in your culture, and the cells engulfs those beads into them through phagocytosis, and the cells are basically treated with LPS. It's one of the endotoxins, bacterial endotoxins. So you are nothing but stimulating this microglial cells to improve or increase their rate of phagocytosis, and that's how you see many of those beads are taken up by the cells. So using these images softwares, you would like to test like how many beads are there in each cells, how many cells are there, what are the total number of cell populations which are taken up the beads, and what are the number of cells which didn't take up the beads. So many questions you can ask. And ImageJ can you can potentially use those softwares like ImageJ to answer those questions. So now in next couple of slides, I would like to show the brain images from my transgenic mouse for Alzheimer's disease. These are 5x FAD, where like 5 different types of mutations are there in the brain, and as expected, they will have the beta amyloid depositions also. So this particular section is coming from the mouse brain, where we have stained these images for glial fibrillary acidic protein, which is an astrocytic marker. So you see those elongations of those cells, and this extension, those bodies are there. So how beautifully they are captured by this GFAP staining, and those small dots are basically coming from different layers. They are also part of the cell bodies or the extension, but they are not coming into the same layer, and that's why you see only those small parts. Now let's say I want to analyze that how much total cell area are covered in this particular image. So what I did is I used one of the parameters in ImageJ to convert that image into a kind of a binary image, and then give me a kind of a percent area covered by those particular cells. So this is the percent area, which shows like 4.445% of the total area covered by these GFAP positive cells. Now, if you want to compare between your control versus the drug treated animals, now let's say my Alzheimer mice are not treated with any drug, and then another Alzheimer mouse treated with a drug, and now I want to see whether there is any effect of the drug on the GFAP expression, and I would like to see that is there any percent area reduced by GFAP, because generally GFAP will be activated in the Alzheimer's brain. So we want to see whether they have reduced down or not. In this particular slide, would like to show that the congophilic plugs, and you see all those red dots are nothing but the congruent positive amyloid beta plugs present in the mouse brain. And once again, so easily you can count, and also percent area can be measured by this images software. Now here is the count, the 45 numbers. You can see like all those numbers, you can count them together, and then eventually that gives you a kind of a total percent area covered. Once again, the similar question you can ask, like how many plugs number, whether they are getting reduced by the drug treatment, or whether by number, or whether they're getting reduced by size. You can ask those questions as well. So now this is a kind of a stepwise demonstration, like how I do it. Now let's say that this is the original image of GFAP. You have to open that with ImageJ, and then convert that into a binary image, into the into black and white, and then you add certain parameters there, like you already have certain inbuilt thresholds in the ImageJ. Like there are 16 different thresholds that are already inbuilt there, so you have to choose which one works best for you. While doing so, you see in this particular image, I have applied all, and that's how you see all the 16 different channels are present in a small kind of a tiles. And from there, you have to identify which particular threshold is actually working for your image. So I see that ISO data, which is here, is somewhat representing my original figure. And that's why I choose the ISO data filter. Now, after choosing the ISO data filter, this is the original one you see. And when you compare between this and this one, they look like pretty much similar, you see. And then you apply the parameters, like what analysis you want to perform on that, like the size in between 20 micron to infinity. Like we know that the cells small drops the sizes are also not going below 20 micron. And in a way, you want to eliminate if there is any, background stains there in the slide. And also circularity and the pixel units, and eventually you apply outlines here. And sorry, and this is what you get here. If you can see that entire things are being outlined, and eventually through the analysis it gives you that entire quantified values. So this is a kind of a protocol. I'm not going to explain you here. I just put them for your reference. You can always go back to this slide and use the same protocol for doing so. But yeah, this is how it is done. Generally, you convert the image into a binary image, and which is like 8-bit image, and then eventually you get those values from those quantified data, and then you want to compare those data between your control versus test animals. So I think this is my last slide. And to just conclude, at the initial stage, as I already mentioned, that image manipulation, yes, it is acceptable to some extent, as long as you do it in a generalized way for all of your image. So now, for every adjustment that you make to a digital image, it is important to ask yourself, is the image that results from this adjustment still an accurate representation of the original data? If your answer is yes, then you are good to go. But if your answer is no, then definitely it falls under the ambit of research misconduct. So one has to be very, very careful when you are doing certain analysis and certain changes in the parameters in images. And while doing so, but because there are lots of powerful image analysis softwares are there, so you have to be extremely careful how you are choosing them, and whether you are choosing in a similar way in between your all the test samples. So I think with this, I will like to end my presentation, and I welcome questions from you guys. Shall I stop sharing?
Yes, sir. Thank you, Avijit, sir, for sharing your insights. And it's a very touching and very sensitive topic in research. Manipulation, either in fabrication or falsification, is unacceptable.
Right.
So now we will take some question from the audience, but before that, I want to ask you one question. So,
Sure.
are there some measure one can implement in the lab to prevent such manipulation by the students?
Yeah, that's why I told you. You see, you should have a curated protocol. Like, let's say that if I have shown you at the last stage of my image analysis, that I have done certain pre-analysis test run to identify that in between 20 to infinity will be my appropriate test size to identify the cell bodies. Now, you can certainly manipulate there, right? Like for your test samples, you might change that 20 to 50, or in your control samples, you would like to say, like, you can change it from 20 to 10 to include many, or include less number of cell bodies. No, you should not do that. And that's why a standardized protocol SOP should be there. And that's how a protocol initially, once you have developed by testing certain number of times, and that is working fine, absolutely all right, and that's when you apply the similar protocol without changing anything to your entire pool of samples. So yeah, so one has to be extremely critical when choosing the protocol, and at the same time utilizing that protocol for the entire population. So that is one of the keys how you can reduce or maybe eliminate the misconduct.
Okay, thank you, Vijit sir. We have Manjari with us, and I think she wants to ask something. Manjari?
Yes, Avijit sir. Very wonderful talk and very insightful how to understand these misconducts. But I wanted to ask in the quantification of immunohisto figure. So when you showed that there is one layer of cells and there are some small dots which are coming from the beneath layer of the cells.
Right.
So while calculating the area, so we have to eliminate those small dots, or we consider it.
No, we'll consider it, because you can well do that. If you want to count only the cell bodies but not the cell extensions, you can do that. And that's where you have to set up your parameters. Like when I have set up from 20 to infinity, you might have to go all the way, let's say 100 to infinity, and that's how you eliminate those cell extensions. As long as you do and maintain the same parameters in between your control and test samples, you are good. You are in a good shape. But generally here, in a single layer, you will always find so many different cell bodies and also the cell extensions. So you would like to see that overall, how much area is covered by the cell bodies or any portion of the cells in that particular layer. So I would not, like to eliminate any of those dots, because I know those are coming from other cells which are probably below that layer, and only the extensions are, identified in that particular layer.
So in continuation with the same question, in the whole section, the area is calculated for whole section, or you choose a particular area only, or
No,
3-4 places.
no, I choose the whole area. Basically, the entire microscopic view what I have captured through my microscope, I apply the same area for all the images, because when you are trying to concentrate on a single or like 2 to 3 areas, you have to be extremely careful that you are not, reducing or increasing the area size. But you can well do that. Like, let's say that in a 5x magnification image, you want to exclusively look at like dentate gyrus or CA1 or C3 regions of the hippocampus, but not under the, under the cortical regions in the brain. You can well, choose your area. But once again, what you do is you normalize that from the total area, and you follow the same pattern in between your samples. You can do that as well.
In the published data, it can be very difficult to identify, because generally a section of the area is published, right? So how to identify that the area which they have reported in the publication is for the whole area?
I mean, generally, you should clarify those into your research methodology. I mean, when you are writing that how you have analyzed the images, you generally, mention that, okay, we have taken that image and we have taken that entire area, or if not, that we have specifically, concentrated on the dentate gyrus region of that brain sections, and we use the similar pattern for all the sections together. So you basically have to mention that either into your methods or into your, image legends.
Okay, sir. Thank you. Thank you so much.
Yeah.
Any other question, Radhika? So meanwhile, I got one question from Facebook from Rita Isaacs. She is asking, how do neurons affect behavior and which part of brain controls social behavior?
I see. Yeah, I mean, see, the brain is a most complex structure in our body, right? So, and they are well networked and well versed with different regions. So for the social behavior, obviously, the hippocampus is one of the major sites where you have this special and, working memory being I mean, controlled by that regions, and also the part of cortical regions are also, plays very important role. So it's like always a complex connection between the cortex and the hippocampus will have a certain role in this memory formation and social memory formation as well. So like when I say the social memory or social behavior is concerned, they are always mostly associative memory, and the associative memories are also, linked to the hippocampus. Now let's say that if I give an example, that if I ask you what did you have in your breakfast last Saturday, you might not know, you might not remember that. But if I ask, like, okay, fine, what did you have, I mean, what food you had on, let's say, my marriage or in somebody else's marriage, you will certainly remember that those are the menus you ate during that day, because you have an associated connection with that particular food to a certain event. And that's how you actually, that helps to convert your short-term memory into long-term memory, being associative. And that's how it is well networked and well connected. And it's very complex to specifically mention that, okay, this is the very same region, helps in consolidating memory, social memory. It will be very difficult, but yes, hippocampus plays a major role there.
Okay, so anyone have any question can ask? Okay, I think we don't have any questions. So thank you, Avijit sir, for coming. It
Welcome.
was a wonderful
Me,
talk.
sir.
Okay.
Is
Yes,
there any
Priya.
question?
Yes.
Hello. Good evening, sir.
Hello, good evening.
So my question is, we are using BSA as a blocking serum
Okay.
in IHC. So it affects our result that if we are using BSA from BSA use from raised in the goat or rabbit species. So and I am dealing with mice model, so it will affect my accuracy.
You mean in your mouse brain sections, you are using BSA to
I section, sir.
okay, I section, you're using bobenzin and albumin to block the sections from
Yes,
any
sir.
non-specific bindings. See,
Yes.
these are very, very subjective and qualitative. I really cannot say that which works best for you. It's all, lab specific, and even sometimes I have seen person specific also. You are using the same protocol, it works with somebody, it doesn't work with somebody. It's all depends upon how you are using. So if BSA doesn't work, you can go for 5% whole milk, but make sure that the milk is thoroughly dissolved into PBS, and then probably stain it through the syringe filter so that it doesn't blot into your sections. And some of the other way you have to try and figure out which particular protocol works best for you.
So in some papers, some of the authors have used secondary antibody with secondary antibody raised blocking serum.
You mean the blocking serum for the secondary? Yeah, that should be the general approach. Now let's say that your secondary antibody is raised in goat, then you use goat serum for blocking the samples,
Yes,
because
sir.
that's where you don't want non-specific binding to happen. Because let's say that for overnight you keep your sections with primary antibody, and you know that anyway my primary antibody is too specific and they will go and bind there, but eventually now my secondary antibody will be too strong for the signal, and I don't want that to go and bind to some other regions also. So that's why I want, how I can stop or maybe at least reduce it, use that particular serum where that particular secondary antibody is raised, so that all the IgGs, open IgGs, will be blocked by that particular serum in your samples, and then you are putting your secondary antibody. And even yes, even you put your secondary antibody also into that serum. I have seen, but it all works. Sometimes, like let's say your signal is very robust, you have got a very high chance of non-specific binding. You want to include couple of more steps of blocking so that you don't want non-specific bindings. But sometimes your signals of that particular antibody is probably not too strong, so you don't want too much blocking so that your secondary antibody also don't find the place to go and bind to the primary antibody. So that's when probably you don't want to use your secondary antibody mixed with a blocking serum. So I have used both sometimes, and it's all subjective. Like let's say your GFAP might work there because that particular signal is abundant, but IBA1, which is like the microglia marker, that may not be as robust as GFAP. So you want to tweak around your protocol. It's not necessary that the same protocol will work for your all different proteins.
Okay, thank you, sir.
No problem.
Anyone wants to ask any question? Um, Thank you, Avijit sir, for wonderful presentation. We are always excited to listen you and to have your experience. So thank you very much for coming, sir.
Thank you. Thank you for calling me here. It's really wonderful. I truly miss NRL, and I can see few of the faces from NRL. Like I personally know Parul, and so glad that she has taken over my research question, what initially we have formulated, and then carried it further. So it gives extreme pleasure that when you have uh, worked on a particular research question and it's not the end of the road. Through Parul's work also, we got a couple of publications, and that's the way it should be. You ask a question that lead to the another question, and then eventually you go forward. And I'm really glad that I'm still in that same background. I'm still working on Alzheimer's. That's my thirst area. And in this particular lab, I am working on the drug discovery program. So it's really amazing to see that how exactly the drug discovery program works from the bench to all the way to the clinical trial levels. So initially, there are chemists in our lab who synthesize those drug molecules, and it's not that easy. Like that one drug you create, and eventually that will end up, using for the animal testing. Probably out of 500 drugs, you get hardly 3 or 4 drugs, which probably will pass through or cross through all the pharmacokinetics and end up using them for the treatment in animals. And then in the animals, if you find something, then only it moves to the human. So yeah, out of our work, I am happy that recently our lab got NIH funding for one of the phase 1 clinical trials for one of the candidate molecules from our lab. That's basically tested in Alzheimer's mice and also in epileptic mice in our lab, and carried forward to the clinical trials. So it's a good way to show that how exactly from the drug development point of view, how it moves from synthesizing the molecule to all the way to the testing in the cell or in animals.
Oh, that's very interesting, sir. So you are diving very deep in the field of Alzheimer's disease.
Right.
Well, thank you very much, sir. Thank you, viewers. Thank you very much. Goodbye, all.
Thank you. Take care.